Since I'm not in class today with you, I wanted to make this so that you could help understand the process of chemical reactions with enzymes, what those are. We'll go over these together on class on Tuesday. But essentially, if we remember back to chemical reactions, you learned that the things that you start with here on the left side of the chemical equation, those are your reactants. The things that you end up with here are your products. Anytime there's a chemical reaction, it involves breaking chemical bonds, and generally it involves energy. So one example of an important chemical reaction has to do with the breakdown of carbon dioxide in your body, where it reacts with water to form carbonic acid. That's how you're able to get rid of the carbon dioxide in your bloodstream. So if we look at energy, if you remember back to what we did in lab this week, when we did our first test with sugar, you had to heat that on the hot plate and wait for the water to get hot, and it took some time in order to see the color change. The reason you did that was that you were providing energy in order for that reaction to take place. So energy oftentimes is released or absorbed in a chemical reaction, and sometimes it takes what's known as the activation energy or the energy input to get the reaction started. So for example, again, with those hot plates, you were heating the water, you were providing the energy necessary to make that reaction go. So if you look at the graph here, we start here with your reactants. On the left, you end with the products, but this little hill that you have to go over, that is the amount of energy needed to make the reaction go forward. So the reason enzymes are important is that enzymes help chemical reactions take place faster, at a faster rate, and they do that by reducing the amount of activation energy that's required. So enzymes are referred to as a catalyst. They're something that speeds up the rate. So here's an example again. So on this graph, on the left-hand side, you've got your reactants. Here we've got the products. Again, the size of the hill tells you how much energy is required to make the reaction happen. Now you've got two lines here. One's a red line. That's with the enzyme present. The blue line is without the enzyme present. And so what you should notice there is that that red line is lower. That means there's less energy required. That means that reaction is going to happen faster than the blue line. So not having an enzyme present makes it harder for that reaction to take place. Again, here's another representation of that carbon dioxide reaction that takes place in your bloodstream. So the reaction of carbon dioxide with water to form carbonic acid would happen, but notice it would happen so slow that you would have a buildup of carbon dioxide in your blood, and that would cause serious health problems. So without this enzyme, here's the enzyme carbonic anhydrase, that helps speed it up much faster to keep to maintain that stable condition, the homeostasis. So one other thing to know about enzymes is that they're very specific. So one enzyme only works for one chemical reaction. So we learned about lactose intolerance a little bit. You looked up some information and you saw that lactose intolerance is caused by a missing enzyme, the enzyme lactase, which is needed to break down the sugar in milk, which is called lactose. Lactase only breaks down that one sugar, lactose. It's not going to work on any other materials that your body needs to break down in digestion. And so the fundamental concept here with enzymes is that here's the enzyme, this big green blob looking thing. There's a specific place on there, on that molecule, where it's almost like a notch or a cutout. That's called the active site. The thing that is going to react, so the reactants in your equation, in your reaction, are called the substrates. So the enzyme and the substrate need to come together in order for that enzyme to help the reaction take place. And so what happens, here's your enzyme, there's your active site, here are your reactants or your substrate. So those bind together to form this enzyme-substrate complex. The enzyme changes shape, and when it does, it stresses the bonds in the substrate. That makes it require less energy to break apart. And so when it's done, we it has formed this new product at the end there. That enzyme remains unchanged in this process, and so it can be used over and over again. So again, the site on an enzyme, here this little area here, is very specific in its shape. The substrate that binds to that, remember again, the substrate is the reactant, whatever is going to be changed in some way, is very specific in shape to match this active site. So the analogy is kind of like a lock and key. You can't use the wrong key to unlock the front door of your house. You can't use your car key, right? Because the lock and the key are matched together perfectly. Same thing with an enzyme and a substrate. An enzyme is specific because the shape is specific to just that one substrate. So there are many different things that can influence how fast this enzyme reaction takes place, or the effectiveness of an enzyme. So temperature is important, pH is important. And so this video, we'll take a look at the process of how this happens again. So again, remember, enzymes are proteins, and what they're going to do is take a substrate, here's your substrate, your reactant, and try to break that down. So enzymes, you looked up, are really important in digestion, breaking down materials. Here's our enzyme. This little notch right here, there's your active site. That is where the substrate is going to match with the enzyme. So again, there's the active site. And so here's the substrate. As it comes in, you'll notice that the shapes match. So really specific lock and key kind of configuration. So an enzyme only matches that one substrate. So as the substrate binds to the enzyme, we call it a substrate enzyme complex. That enzyme undergoes a slight shape change, and that stresses this bond right here. When that bond is stressed, it requires less energy then in order to break those bonds, because that's the whole purpose of an enzyme, is to speed up the reaction by reducing the amount of energy required. So again, there it's able to break those apart. Coming in as the reactant, what goes out is the product. But notice, although the substrate changes, the enzyme does not. The enzyme is used over and over and over again, and one enzyme can be used thousands of times in order to break down a substrate. Eventually they wear out. Your body does produce new enzymes. It doesn't last forever, but that's the process. The enzyme gets used over and over again to break down a reaction. All right, and that concludes this recording.